Well, the first curious thing was an Italian newspaper that rang up very early in the morning and asked me what I thought about it, but I had to say I had no such knowledge. And then later in the morning, someone from Sweden rang up, or apparently from Sweden, and that appeared to be the right thing, but you know, we're always trained as scientists to question everything. So the first thing you think, well, is this a hoax or a guess or what? So one talks for a while until it seems probably that the person is the correct person. So you begin to think it may actually be right. And then you check a few more things, and finally you then discover when it's announced publicly that it must actually be correct. So that was the feeling on the day of the announcement. Well, I was extremely fortunate to be awarded the Wolf Prize some time ago, and more recently the Lasker Award. And those are very highly regarded awards. Of course, the Nobel Prize is kind of preeminent, at least as judged. If you take the man in the street, they may have heard of a Nobel Prize, they will not have heard of others. So it's a preeminent recognition. And the major sensation you have is that there are people who are highly qualified, very successful scientists who are asked to judge these nominations. And if they consider you're worthy of some recognition, that's immensely rewarding and encouraging. So one's highly gratified if one is lucky enough to receive any of these kinds of recognitions. It's probably important to just tell you what the question was that the research was trying to answer, because one always should start with a question before you try and say anything at all. And the question was whether the different cells of the body have the same genes or they don't. And at the time of this work, that was an entirely open question. And indeed, in the 1800s, it had been indeed suggested that cells might not have the same genes. And the purpose of the research was to find out if that is true. The idea of the research was to take the nucleus containing genes out of a specialized cell, in this case an intestine cell, and to put that nucleus into an egg which had no genes of its own. So you're asking about the combination of an intestine cell nucleus with a non-nucleate egg. And the concept was that if that combination could make an entirely normal adult animal, then that must mean that the intestine cell has all the genes needed to make everything, every other kind of cell. That was the key experiment, and the conclusion, as you may know, was that that seems to work, at least to a good extent, and hence the conclusion was that different kinds of cells of the body do all have the same set of genes. One should know that since the 1800s, frogs have always been the material of choice for embryologists, people understanding development, largely because the eggs are very large and very easy to do things by manipulation, by hand. And so either frogs or salamanders, newts were the choice. And indeed, Spemann, who is very famous in this field, did his Nobel Prize winning work with frogs. So it was the standard kind of material to use, and that was what I was encouraged to do, because there was almost no work at all on mammals at that time, and the mammal embryo is inaccessible, small, and difficult to deal with. So frogs were the obvious choice. The interesting point was that they were using frogs that only produced eggs 2 months of the year, for 2 months of the year. So they had to do their experiments frantically in March and April, and then spend the next 10 months thinking or cutting sections or doing something, and then think of next year's experiments. So my supervisor had the wisdom to say that this work that we were going to do should be done on Xenopus, a South African frog, which has the property of being able to lay eggs throughout the whole year. So I was given the problem of trying to convert the work done with European frogs onto the South African frogs, and that was technically quite demanding, but it worked. I need to explain that the people who pioneered this technique of moving the nucleus from one cell into an egg were called Briggs and King, and they published a very important paper in 1952 showing that they could indeed do that. And when they did that, as long as they started with an embryo cell, that embryo cell nucleus could combine successfully with the enucleated egg. However, when they tried a few years later to do the same thing with the nucleus of a specialized cell, it didn't work at all satisfactorily. It couldn't be done. So they concluded that there must be something changing as cells differentiate. Either the genes are lost or permanently inactivated. And when I came up with my work, a few years later, there was natural skepticism. You know, why should a starting graduate student contest the view of these highly respected and established scientists? So there was indeed considerable skepticism or doubt about whether my experiment was valid or not. So I'd read the paper by Briggs and King and was expecting to get the same result. And to a certain extent I did, but in most respects not. So it was a surprise, but that made it all the more fascinating. If it was possible that the correct conclusion was not what the dogma said, that's a real incentive to press on and see what really is the position and are your experiments really right. I need to explain that the consequence of the experiment I've just mentioned was indeed to make clones, or a clone, and indeed we did that. You have an animal with 20 or 30 clonal offspring, as they're called, and they're like identical twins. And later, using a color marker, it was very clear. Now, the fact is that the experiment that I was doing had really nothing to do with cloning. That was irrelevant. And you will know that it's totally normal practice now to use cloning to propagate trees. I mean, millions of plants are generated for sale by cloning. Now, in animals, that can't be done that way, but it was a side consequence of trying to answer this fundamental question. And since cells do seem to have the same genes, it's possible to make a clone if you spend long enough doing the experiments. So it seemed to me, it attracted a lot of interest, there's no doubt, curiosity type of interest, but nothing scientifically of great interest. The key thing that he and his colleagues did was to show that you do not have to use eggs like I had done to get this reversion from an adult to an embryo state. They were able to take certain gene products or genes, 4 of them, and switch an already growing cell backwards from a specialized state to an embryo state. So Dr. Yamanaka is polite enough to suggest that our experiment influenced his decision to follow that route, but technically they're really quite different. And I think the contribution of our work was primarily to show that different cells do have the same genes, and therefore, in principle, this idea of rejuvenating from adult to embryo cells is possible. And secondly, that eggs at least can do it reasonably well. But in other respects, his approach was entirely different and immensely valuable because he can achieve this without having to use eggs. Now, in our case, we cannot use human eggs. They're virtually unobtainable. So our approach or a kind of design of experiment is inappropriate for any kind of human use, whereas his is entirely suitable for that. I would say yes in 2 respects. One is rather immediate, and the immediate one depends very heavily on Dr. Yamanaka's work, where if it is possible to obtain cultured cells in the laboratory that represent a human disease, such as, say, Alzheimer's or any neurodegenerative disease, then you can test any number of drugs on these cells to find out if certain drugs appear to be able to alleviate the disease. Now, that's of great importance because you cannot legally or ethically inject a whole lot of untested drugs into human beings. But if you have cells in culture, you can indeed do that. So that I see as the first clinical, if you call it clinical, or at least therapeutic application of this work. And the second one is rather closer to the work that I've been doing, and that is in the area of cell replacement. And the concept here is that you take some cells, such as skin, from a human and reverse the differentiation of these to make embryonic stem cells, as they're called, grow those, and then you can switch those embryonic stem cells into a whole range of other kinds of tissues. So you might be able to switch a piece of skin into something like heart or pancreas cells. Having done that, you can, at least in principle, give people back these new kind of cells of the kind that they need, of their own genetic constitution, and therefore avoid the need for immunosuppression. And that would be a great advance, to be able to give people spare parts without having to have them immunosuppressed. As far as clinical trials are concerned, my understanding is, and it's not our work, that this is very close to being undertaken in the case of eyes and restoration of vision. But let me explain that. It turns out that you can make the kinds of cells that do not function properly when people suffer from things like age-related macular degeneration, from which they eventually become blind. Now, you can make these retinal pigmented epithelium cells, which are support cells for the photoreceptors, and indeed photoreceptors in culture. And to my amazement, it's possible for people to inject those cells into the back of the eye, and they will then integrate and actually function in the eye into which they have been injected. And my understanding is this has already been successful in animals, giving some degree of restoration of vision, and there's no reason that I can imagine why this shouldn't work with humans. And I understand that they will be starting human tests on this next year. Yes, I didn't have, at the time this work was done, any feeling for implications relevant to human survival. Remember, this technique had been made to work with frogs, and a long time later, 1980, there were papers published in a highly regarded journal saying it is not possible to do nuclear transfer in mammals, in mice, cannot be done. And then much later, Ian Wilmot and his colleagues were able to show that it could be done in sheep. So that really opened up the whole of this kind of approach to having some potential relevance for mammals and indeed humans. But I would say until that time, I didn't see any obvious therapeutic value to the work. Yes, I think it's a very good question, and I'm particularly sensitive to that because we all have to make a good case as far as we can for receiving funds to do our research. And increasingly we're being asked to say, what value will your research have for human health? So we're always being questioned on that, and we try to do as well as we can. But it would be a great pity, in my view, if work that cannot immediately be justified as being likely to relieve human health is excluded. And this example of the work I've been able to do is, I think, a particularly good one, where done in the 1950s, 60s, with no therapeutic value, wait a considerable amount of time, and the potential value emerges. So my general view is that any basic research that is going to tell us more about how cells and their components work will eventually turn out to have some therapeutic use, even if that's not evident at the time. So I hope that this will be kept in mind by those who judge the distribution of funds. I think the limiting factor, there always is a limiting factor in everything, must be funding. Without funds, you cannot do these things. Even simple experiments are expensive. So I will put that as number one. And I think there is a significant constraint, in my view, due to regulations. And the so-called FDA issues all sorts of regulations about what may be done to give humans some benefit from therapeutic advances. And the view I'd like to express is that if someone is really very unwell, they should have the choice as to whether they will receive some therapeutic treatment, not the lawmakers, priests, etc., etc. If you tell someone that something might be helpful to them, and this is what you know about it, surely they should be the ones to take the decision. And I've known someone for some years suffering from very serious Parkinson's disease, and they've been suffering from this for a long time. And their line has been exactly that. I'm quite old, they say. I'm suffering a terrible deterioration, health, everything getting worse. Any hope at all is worth it. So it would to me be crucial to try and transfer this whole decision process to the patient away from lawmakers. Wow, well, I suppose I've lived 50 years. I won't live another 50, but it's amusing to think, can one guess? So I would volunteer the view, probably quite wrongly, that within 50 years we will understand everything about how cells work and how embryos develop, except possibly for how the brain itself works. I can't imagine at the moment how memory can operate. It must be a way, but I don't see it. Whereas to go from an egg to an animal with the normal structures, I think we must surely understand that fully within this time. Yes, the context is that at that time, and remember I'm talking just a couple of years after the end of the last World War, 1947-ish, we didn't do any science at all until you were age 15. At that point you had your introductory first term lesson in biology. And I was bottom of the bottom class, bottom in the whole year in achievement in biology. So I was worst out of 250 students at the time. So they put on a relatively inexperienced teacher. In fact, I think he wasn't a teacher at all. He's probably a museum curator. And the problem was that he used to dictate to the end of the room. He'd sort of look at the ceiling and dictate to it. And I was no good at taking notes. And there were no textbooks. So you'd go away and then you try and read your notes and remember what he might have said. The next lesson you come in again, look at the ceiling and say, get out your notebooks, 50 questions, question 1, 2, 3, up to 50. I couldn't answer any of these questions. So he comments that I scored 2 marks out of 50, I think, on one such test. But I couldn't handle that kind of teaching. And so he gave this crippling report. You may have noticed things like any thought of Gurdon being a scientist is completely ridiculous, and this would be total waste of time on his part and on those who would have the job of teaching him. So that terminated completely my whole science training at that point. No more science for you, they said. That's clearly, you're hopeless at that. So I did something else like ancient Greek and Latin, wasn't quite as bad at that. The fact is I couldn't for the rest of my school time. I was put on to ancient Greek and Latin, and then my parents, particularly my mother, could see that I was really most interested in insects in particular. All my free time was spent actually reading insect books, not Greek and Latin. And so she then arranged, through a number of connections, with the head of zoology in Oxford, to enable me to do 2 extra years training how to start science at 18 and then get far enough on to be able to do a university course in it. And it was agreed with this nice man that if I could survive those 2 years, they would let me into the university course in zoology. And it was very hard work. I have to say, very hard work to pick all this up quickly, and it happened to work. So I was able to start the course, and that then got me into doing science. So I was extremely lucky to have that opportunity, and really thanks entirely to my parents, otherwise it couldn't have happened. And then life really started when I became a graduate student with a wonderful supervisor. But the lessons are amusing. In the newspapers I read now, they've really taken notice of that and quote a number of people who've had somewhat similar experience. But I warm to their view, which is that if a teacher thinks a student's bad, the student shouldn't be too discouraged. You should try it, and if you're really interested in something, keep going. Don't give up just because the teacher doesn't think you're much good. From a very early age, I just was fascinated by animals and plants, and I always used to think, how can it be that a seed grows into a plant? It's an extraordinary thing. How can it know how to do it? And that fascinated me, whereas I wasn't terribly interested in being able to translate the Iliad or Odyssey. I mean, people can do that anyway, but the biology is, you're finding out something new that isn't known before, and I would not have liked to spend the whole of my life getting back to where someone was already. You like to go forward, not backward, and that's the great appeal, I think, of science.